We've talked about how enzymes catalyze reactions and how an inhibitor can impact an enzyme's function, but now we need to talk about other ways in which enzymes can be regulated. So first of all, we've already talked about allosteric proteins in the context of hemoglobin. We talked about allosteric protein, but allostery is also a phenomenon that enzymes can observe. Generally, allosteric enzymes are going to have two subunits, or sorry, multiple subunits, and those allosteric enzymes will ultimately take on a pair of different forms or kind of shapes. And those are commonly known as your T and your R state. And those are exactly what we talked about with respect to hemoglobin as well. You have your T state and your R state, your tense and your relaxed state. Now, what ultimately that means and what's displayed right here is an example of an enzyme in its inactive form or its less active form, and an example of molecules that can play a role in either activating or inactivating an enzyme. So here we have the general concept. On the left-hand side, I'm going to put a little star by it. We have the general concept of a less active enzyme can bind a positive modulator. So a positive allosteric effector. Allosteric effector. So whenever that protein binds to that positive allosteric effector, what's going to happen is that protein is going to undergo a change or a structural change which enables a substrate to bind. So we already kind of talked about this with respect to proteins and homotropic and heterotropic interactions. So we talked about bisphosphoglycerate. We also talked about O2 as a positive allosteric effector of hemoglobin. Now here we're just presenting it as random positively or positive molecule. When it binds right here, it's going to cause our what was previously a rough rectangular shape to now it has some different angles to it. Whenever it undergoes that conformational change, now all of a sudden our native substrate can bind. So this is displaying a heterotropic effect because of the fact that the protein or the molecule that binds is different from our native substrate. So then we've got our model right here where we have our enzyme that has bound its positive modulator and its positive allosteric effector, and its actual substrate. Now over here, what we have is, on the right-hand side, which I'm going to indicate with a smiley face, we've got, there we go, we've got a rough representation of a metabolic pathway. Threonine being metabolized into isoleucine. Now the first enzyme in that pathway is an enzyme known as threonine dehydratase. Now what this pathway is showing us is that we have kind of a negative allosteric effector. Isoleucine comes up here to inhibit the function threonine dehydratase. So think about that as a negative feedback loop. Whenever you've generated your product, well, that can go back to regulate something or downregulate something upstream so that you do not have that continuously going on. So that's kind of allosteric. So you have these two different forms, and that's kind of a hallmark of allosteric enzymes and allostery in general. So in addition to enzymes that bind to small molecules, that those cause some conformational change in the structure of the protein, we have something known as covalent modification. And that covalent modification is, as an example, adenylation or methylation. We're going to talk about some other examples on the next slide, but these are two pretty common examples. And what this consists of is, and I'd like to draw your attention to this box right here, which I'm indicating with a star. What we see here is a reaction in which an enzyme reacts with ATP, releases PPI, or inorganic pyrophosphate. So that is, yeah, those are the last two phosphate groups on that ATP molecule. What remains behind, that AMP basically, well, that gets added to your enzyme, okay? Now, what's important about covalent modifications is that they are reversible and they are specific to individual amino acid side chains, okay? Now, this is generally going to happen to a tyrosine residue, hence why we've got TYR in parentheses there. Okay, now another example of a regulation or a modification that regulates an enzyme is methylation. So methylation is a post-translational modification, which we've already talked about post-translational modifications. Um, but that is an example of a modification to a glutamic acid residue. We have our enzyme, we have a methyl donor known as S-adenosylmethionine, and what we're going to do is we're going to add simply a methyl group onto our enzyme. Now that S-adenosylmethionine is going to donate our methyl group. That's going to become S-adenosyl homocysteine. That methyl group gets added to our enzyme. Again, this is being added to a specific or a glutamic acid residue. And what these modifications are going to do is they're going to take either an active enzyme and form an inactive one, or it's going to take the inactive and produce an active form. Okay, so I think that it's helpful to keep those things straight. These are just two examples of covalent modifications. Next up, covalent modification that I would argue is maybe I would say it's the coolest or the best or the most fun, and that would be phosphorylation. So phosphorylation is the addition of a single phosphate group. Now, what I'd like to draw your attention to are your donors. You've got ATP coming in along with your enzyme, and what is released is ADP. So ATP, adenosine triphosphate, and you release adenosine diphosphate. What that means is that the last phosphate group on that ATP molecule gets transferred to our enzyme. So we have our phosphorylated enzyme here. Now there are specific residues that can be phosphorylated: tyrosine, serine, threonine, and histidine. Well, those first three, threonine, tyrosine, and tyrosine, serine, and threonine, all of those have hydroxyl groups. Those hydroxyl groups serve as great places to put a phosphate group. Histidine does not, but that doesn't mean it cannot be phosphorylated. Okay, now what we've got here is an example of a less active enzyme known as phosphorylase B. When you go on to take biochemistry 2, you'll learn a little bit more about phosphorylases and phosphorylase kinase and enzymes that are involved in activation of metabolic pathways and everything like that. But phosphorylase B is inactive whenever the serine residue number 14 on its dimer, well, it's a dimer, so it's two identical copies of the protein that dimerize together. Each one of them has a serine at residue number 14. Now, each of those serine number 14s are not phosphorylated, okay? An enzyme known as phosphorylase kinase. Well, that is going to transfer two phosphate groups from two ATP molecules. Well, it's going to put those phosphate groups on both serine number 14s on each one of those proteins that make up our phosphorylase A dimer. Okay, that will make an active form of the enzyme. Now, one thing that's worth noting is just because something gets phosphorylated does not mean that it is the more active form. In many cases it is, but not across the board 100%. Okay, so then we have another enzyme that kind of regulates in the opposite respect, or in the opposite direction. Phosphorylase phosphatase. What that does is it removes these these phosphate groups from serine number 14. That will produce our less active phosphorylase B, our dephosphorylated or doubly dephosphorylated phosphorylase B. So it's important to know that modifications, and this I think really is a great example of the reversible nature, because this is showing the two different enzymes that are involved in the activation and deactivation. Phosphorylase kinase, well, that's transferring a phosphate group. That is activating our enzyme. Then we've got phosphorylase phosphatase. That is a phosphatase. Phosphatases remove phosphate groups. That is removing those phosphate groups to recreate that less active enzyme. So think about this as you do not have to have that enzyme functional and on 100% of the time. Instead, you have this regulatory effort by other enzymes that can either turn on or turn off an enzyme. All right, well, that's all I've got for enzyme regulation. I hope this is helpful. Thank you very much. Have a good one.